So my talk is very much like Sidonia's, almost exactly. We're going to show you trying to kill weeds and they're just blooming. The only big difference is that's faster, even faster than 3 years with the bacteria. And I'm also going to say that I don't do any research on this. I do more environmental work. But I thought this would be more of interest to an audience with a lot of different interests. So we're going to talk about the antibiotic-resistant bacteria natural selection. I could have talked about, put a phylogenetic tree up and talk about the last common ancestor, or I could have talked about cyanobacteria and the great oxidation event, but I didn't want to have to come to geology and talk about rocks. So I'm going to stick with this. And to begin with, just a brief overview. I mean, all of you had microbiology eons ago and probably wish you could, didn't have to think about it again. But where do we find them? Everywhere, of course. And what do they do? And so where do they exist? How abundant are they? How do antibiotics really work? And so based on how they work, exactly how have we had the evolution of antibiotic-resistant bacteria? And at the end, I'm just going to give a brief mention about the superbugs that we're all starting to hear about in the media. So where do microorganisms exist? Well, of course, everywhere. So this, of course, is a mat on a pond, which has some algae, I'm sure, but there's some cyanobacteria. Here's just showing, I think this is an alfalfa root. Essentially, there's some fluorescent probes that are showing bacteria, or Pseudomonas putida in the alfalfa root. Here's the hydrothermal vent, the black smokers. There's bacteria down there growing. These are bad bacteria, I guess, if you want to think about it. Here's our termite. Well, our termite cannot eat wood, essentially, on its own. It has to have bacteria to digest the cellulose. And then the ones that are kind of scary, and it's good that they're actually friendly bacteria, they're ones that grow on, essentially, nuclear rods. So they grow just fine. They can repair their DNA. It's a good thing they're not pathogens, though. And then here we're looking at, so antibiotic resistance. A lot of people don't think about where most antibiotics come from. We have some synthetic ones now, but this is a Petri plate showing essentially a sample taken probably from the soil, diluted out. These areas here that have clearing around them, these are actually bacteria that are producing antibiotics, and they're killing off their neighbors. This is what everybody, the non-environmental microbiologists think of, though, the pathogens. And I've tried to keep the nasty pictures to a minimum. But here we're showing streptococcus. So you have your strep throat. Here's essentially a Petri plate, and this is sheep's blood agar. And so you can see the area of clearing. This bacteria essentially is lysing the red blood cells. And then this is your if you remember from your general micro course, you streak out E. coli, you get this on the special medium, you get this nice metallic green. But this is what the food poisoning comes from if you eat the contaminated meat. And then just a little numbers here for abundance. So these numbers are probably, hopefully, close to accurate. The population of Illinois, we're nearing 12.9 million. The U.S., we're at 300 million. Earth, we're at 6.9 billion. And then estimated microbes, what's 10 to the 30th? A nonillion or something like that. And I think that might be accurate. I think I might have 30 zeros there, give or take. And so this is just one thing that, is kind of interest. If you think about the estimated population of microbes on Earth, if each microbe is essentially 1,000th of a millimeter in size, and we line them up end to end, they would span the distance from the Earth to the Sun 200 trillion times. So there's a lot out there. And they're super tiny. This is just showing bacillus cells on the tip of a pin. Another thing to think about is us and microbes. So they're the most abundant life form on Earth, but just think about how many we just have in our intestine. So 10 to the 14th microorganisms, and this is just showing the different region of the intestine and the type of bacteria that are normally present. And then if you look at this, the amount of bacterial cells per person, 70 times 10 to the 12th. So we are literally walking bags of germs, right? I mean, we have 10 times more bacterial cells than we have human cells. The good thing is most of them don't make us sick. But evolution of bacteria today, so this isn't happening just in the backyard in southern Illinois, but this is happening everywhere. So they're the most abundant life form, and they grow quickly. So if you think about how could we evolve to antibiotic resistance.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
kind of scary movie if this is a flesh-eating bacterium, right? Let's go back to the PowerPoint. All right. And like I mentioned, they can thrive in any environment through genetic variation. So this provides capabilities essential for survival. And these genetic innovations arise from random mutation, or later I'll talk about what's neat with bacteria is they actually do have some sort of exchange of genetic information. You could say conjugation is a form of bacterial sex. And if the traits confer an advantage, they are retained. So what about antibiotic resistance? So we started using antibiotics about the 1930s. So sulfonamides were the first ones that were used in the war. Since the 20th century, bacteria have evolved resistance to all antibiotics that we use. So extensive use of these drugs has essentially caused strong natural selection in favor of those mutations, which confer an antibiotic resistance. And today we even have those superbugs that are resistant to multiple antibiotics. And this is extremely problematic in hospital settings. So here's just a table from a textbook showing the year these different antibiotics were employed. So sulfonamides were employed in the 1930s, and when was the first bacterial resistance observed? In the 1940s. Penicillin took 3 years. Anybody have a good guess for this? Where does penicillin come from? Molds out in the environment. Bacteria have already been exposed to it. Things like streptomycin. I think the one I was looking at here, I guess erythromycin, we've had the longest use without antibiotic-resistant strains. Vancomycin, methicillin. Where do you guys hear of methicillin? What's that acronym that everybody hates to hear about? MRSA, methicillin-resistant Staphylococcus aureus. Resistance developed in a year. It's very similar to penicillin. And there's ampicillin and cephalosporins. And so this is a really old figure, but it kind of shows the same thing that Sidonia's graph is showing. We're looking at vancomycin-resistant Enterococcus. So Enterococcus can cause urinary tract infections, gastrointestinal infections, a range of other infections. But here they were essentially documenting these vancomycin-resistant strains of Enterococcus in hospital intensive care units in the U.S. And so we're going along just fine, and then as you can see right about 1990, the incidence starts and it just explodes. So in 2001, it was already up to almost 30%. So the speed is just amazing of how this happens. So the key, this will be your molecular lesson, and I'll leave it really, really brief here. So the take-home message here is, how do antibiotics work? Well, they target molecules essential for bacterial survival. They don't target things that would hurt us. Okay? They target the differences between bacteria and us. So you have things that target DNA replication, transcription, protein synthesis, and cell wall. So how does a bacterium become resistant to this then? Essentially, if one's targeting, let's say, your DNA gyrase to replicate DNA, mutation, if a random mutation occurs in the gene for gyrase that makes it no longer a target to the antibiotic, that bacterium is now resistant. So this is just showing you a different slide, or how bacteria become resistant. And so you can have, like I was mentioning, modification of the antibiotic target. We have everything from essentially picking up a gene from a plasmid, which I'll talk about later, for an efflux pump. So essentially, if the antibiotic goes in, it can just get chucked right back out. Or you can have essentially some sort of enzyme in the cell that will degrade the antibiotic. Or you can alter the antibiotic into a form that no longer hurts the bacterial cell. So there's a range of different ways to become antibiotic resistant. The big one I was just going to go over, because it's the easiest to understand, is just random mutation from DNA replication, essentially. So a single error in DNA replication can result in antibiotic-resistant bacterial cell. And so not for all of them, but for macrolides here, which are used to treat gram-positive, essentially it targets the ribosomal RNA. It's in the ribosome. And so they've shown here that you could have a base change in just one of these 3 regions, and the bacterial cell will be resistant to macrolides. How frequent can this occur? Well, you saw how fast they can grow. So here's some statistics that I pulled up. Essentially, if you had a bacterium that had a genome size, so its chromosome was about 5 million base pairs. You consider each one of these a base pair here. And the average random mutation rate is 2 times 10 to the minus third per genome. Well, you don't want to think about fecal matter, but in 1 gram of fecal matter, there's 10 to the 10th to 10 to the 11th cells. So essentially, in that 1 gram of fecal matter, it's likely to contain a novel point mutation conferring macrolide resistance, just from random change. And so just essentially changing one of these base pairs to something else, if it's in the correct location. So what happens once it becomes antibiotic resistant? So we're not selectively forcing the cell, right? We're not forcing the mutation, we're selecting for the mutation. So if here are essentially your system, and here are your bacterial cells, and this guy here in red essentially had that random mutation that makes it resistant to the antibiotic. He's outnumbered, right? He can't compete. He's just going to either die or just barely get along. But then you take the antibiotic, and what happens here is essentially now we're killing these guys that were sensitive, and the resistant guy is still here. And then next time scale we go, well, the resistant guy has exploded. If you think about it this way, competition keeps population of antibiotic-resistant bacteria in check if no selective pressure. This is a great figure to say don't take antibiotics if you don't need them. So here it's showing essentially in your gut you have these microbes. Here are 2 in red that essentially had a random mutation that make them resistant to antibiotics. Now let's go ahead and take the drug. Well, just like I said, we're going to wipe out all the sensitive guys. Now there's tons of food for these guys to compete with, the resistant ones. And your entire system is going to be essentially all pathogens that are making you sick. What's really becoming a problem now are people are taking antibiotics, and a few unlucky people have certain strains of Clostridium difficile. You've heard of the C. diff infections? You can't get rid of them hardly at all. You go into intensive care on IVs because severe diarrheal disease, till the point till it's going to kill you. Strangely enough, I went to a talk last year where they have a 90% cure rate. And do you know what the treatment is? Fecal transplant. That's 90% cure rate. Why isn't it used more often? The FDA hasn't quite approved that one yet. But yeah, Mike Sadowski at Michigan State, they're doing this. And why do people say, okay, sign me up for it? You've lived in hospitals for years with this. They'll try anything. So if you ever saw that on TV where was it the wife got a fecal transplant from her husband? Usually 3 days later, they're fine. Because essentially you put new bacteria in to help compete against the pathogenic one. Then the other thing, so I think of, I guess as a microbiologist, I think on the micro scale, I think of evolution of pinpoint mutations. But another reason why we get the superbugs and a lot of antibiotic resistance are these plasmids. So these plasmids essentially pick up genes for antibiotic resistance. And a big reason this happens is because, well, a lot of antibiotics are produced by bacteria. Streptomyces, a lot of the soil bacteria produce antibiotics. Those bacteria have to be resistant. And so they have genes in their chromosome or on plasmids that make them resistant to the antibiotic. Well, this now essentially gets transferred to other types of bacteria. And so this can happen from transformation. Essentially, you can just have a cell die in the environment, and it lyses and its DNA is released, and some other cells can be naturally competent, or they can naturally take up the DNA. Another big thing I didn't even talk about are viruses. So there are viruses that infect bacteria. They often can carry genes for antibiotic resistance that they've picked up from killing other bacteria. And then, my picture is not as slimy as yours, Andy, but then there's bacterial sex or conjugation, where essentially they transfer DNA back and forth between a pilus, or between a conjugation bridge, but the pilus holds them together. So what's the take-home message here? Well, bacteria resistant to multiple antibiotics are considered superbugs. And so I didn't bring a plate with MRSA. Didn't think you guys wanted to see that. But any of you know David Clark, who's here in Life Science 3. He wanted me to come and show you his superbug. And he's got a cape when he says MRSA. And if you look at his face, I think David's cat's got it. But essentially, if you're interested, David has every type of fuzzy microbe that you can imagine. But here it's showing essentially this methicillin-resistant Staph aureus has been exposed to tons and tons of different types of antibiotics, and now it's resistant to almost all of them. And I won't even go into the fact that we're using antibiotics in agriculture just to try to keep animals healthy and to increase their growth rate. That's not helping anything either. So it's in the milk and in the food. But what are these superbugs? Well, the original one is the methicillin-resistant Staph aureus. And so according to the National Institutes of Health, roughly 1 in 50 people carry a strain of this that's resistant to these antibiotics. And if the bacterium invades essentially a cut or opening on your skin, it can cause a minor infection like a pimple, or sometimes it can do a serious widespread infection involving the heart, lungs, bloods, and bones. Other things, if you hear of VRE, vancomycin-resistant enterococci, this essentially has become a big problem. The big one in India, and then we were talking about in England now, is essentially the extensively drug-resistant tuberculosis. It's not in the U.S., thank goodness, but they're having a lot of problems treating TB, especially in India. And then the last one is the new superbug. So gonorrhea used to be treated with take a pill, gone. Now it's resistant to pretty much everything. So there are strains in Asia now that are even resistant to cephalosporin. So there's nothing to treat that superbug. So the take-home message that I want to get across is essentially this happens quickly with bacteria, more quickly than anything else, just because they grow faster than anything. Not all of them, but most of them have a fairly short generation time. And it's essentially, don't put the selective pressure on them, or it will happen. So that's all I have.